Thank you very much for this invitation. Indeed, I would like to address the question of unwanted immune responses in gene therapy, and I will focus particularly on gene therapy of genetic disorders, because in this context, you really don't want to have any recognition of your vectors, whether they are viral or non-viral. I'd like to remind a few concepts before going into certain examples and solutions, how we can overcome this problem. The first concept here is to keep in mind that the immune system is a very complex system, and it's constantly a balancing act between cells which have been educated to respond and recognize invaders, but also regulatory mechanisms and a number of different systems which are there in place to recognize pathogens as well as tissue abnormalities, let's say. And so the introduction of gene therapy in this context is obligatorily going to disturb this balancing act. And we're talking here about gene therapy that's injected in vivo systemic administration of vectors, whether they are viral vectors or gene-modified cells, for instance, but also the introduction of nucleic acids in the context of these non-viral approaches. And this is really a major concern, even though I think it's been overlooked for many years. It is now in guidelines for the development of gene therapy products, but it's actually a very complex problem, and it's also difficult to really correctly model, as we've just heard previously. Murine models are not always the best models to in fact study immune responses that will in fact arise in man. So there's no mystery, however, a viral vector, gene therapy vector, will enter the body, and it will in fact be recognized like any other type of immune invader. So it will activate the innate immune system, all of the cells of the innate system. It will in fact be captured by antigen presenting cells and presented to T and B cells to mount adaptive immune responses. And I'd like also to emphasize, because this is very often overlooked, that these immune responses are not only complex, but they are also dynamic. And you cannot understand what's going on if you just look at one time point. So you have to understand that, some mechanisms occur very rapidly after the introduction of the vectors or whatever stimulus. And we're talking about minutes or hours, and this generally deals with innate recognition mechanisms, unless you've had actually prior exposure. But then you prime the immune response and you mount that, and you have to generate first the priming, then the activation of clonal expansion and differentiation into the effector cells after you reach a phase of exhaustion, regulation, and then you establish this long-term memory. So again, it's very important to sample these immune responses at different time points to know in which phase you're going. So all of this, of course, is very dependent on whatever vector you're using, their properties, but also all the pharmacology, and also, of course, the host status. This is a very important factor. And we've heard previously a very interesting presentation of all the microbial associated patterns, such as the toll-like receptors, but they are not the only ones. But unfortunately, these are very clearly present in the tools that we use in gene therapy. We can have contaminants in the preparations, coming from, let's say, microbial contamination with LPS. And this is very often not even checked into preparations that are used for preclinical studies. But of course, you have also signatures such as CpG motifs in the DNA that you use. And also, this is not necessarily taken into consideration. So I think this is something that needs to be looked at a little bit more carefully. We also know that from the COVID crisis that people are not equal in terms of immune responses, and people, it's been very clearly seen that people infected with SARS-CoV-2 can react in very different manner. Some people, in fact, have developed these cytokine storms. So it's very interesting now that there are studies that are in fact ongoing to try to understand and see if we can predict those factors that in fact will determine the susceptibility or the variability between individuals in their ability to mount immune responses. So some of these studies have identified, for instance, by looking at PBMCs in a large panel of individuals, that you have some factors which are non-genetically determined by, let's say prior behavior or by infections, but others are in fact genetically determined. And this in particular concern innate factors. So I think this is still in its infancy, but these are factors that really should be looked much more carefully when people are perhaps including patients in their gene therapy protocols. The other thing that needs to be also taken into consideration is the fact that these innate signals are not only directed against external pathogens, but can recognize also motifs of damaged cells. And damaged cells, generate a large number of different signals that the cells can pick up and then react against, and that can generate a lot of inflammation. And this, of course, will be very variable if you're dealing with the pathologies for which you're trying to apply gene therapy. So, for instance, very inflammatory pathologies such as Duchenne muscular dystrophy will in fact be treated in the context of a very different context than a normal individual, and you will have a lot of dumps and a lot of inflammation that will completely modify your immune parameters. Now, immune responses in gene therapy are very scary. They can be very problematic. And of course, everybody knows Jesse Gelsinger, who was the first death of gene therapy. He was treated with an adeno viral vector a long time ago. And this has completely changed, of course, our perception and of the risk of gene therapy. And this has led people to, in fact, use much less inflammatory vectors, such adeno-associated viruses, which are used now in hundreds of clinical trials in rare or non-rare diseases, and have in fact led to approved products. There's different serotypes. These are very small viruses that diffuse everywhere, but they are using different capsid serotypes that determine their entry and their interactions with target cells. They are DNA viruses, but they're using either with single strand DNA or double strand DNA to facilitate, in fact, the transgene expression. So I'd like to emphasize also that according to different protocols, the dose that's used for AAV in human can be very, very high. And we're seeing now that for systemic administration and the therapy, for instance, in neuromuscular disorders, very, very high doses such as 10 to the 14 viral genome per kilo have to be used to get therapeutic effects. So these are really very strong stimuli that are given to patients. So I'd like to focus on AAV because this is a really interesting drug, and people are in fact using it for gene transfer, but also for other types of applications that may be interesting for people doing RNAi or CRISPR therapeutics. But AAV in general has limitations, and some of these limitations I'd like to discuss. First of all, there's a number of individuals, it depends on the serotype, but who have already been exposed to natural AAV. And so they are responding to these natural AAV capsids, and they have mounted antibodies against that. So there's a pre-existing immunity, which makes them ineligible for gene therapy now, because they have these blocking antibodies against the capsid. And so nothing can pass that. The other problem is that when you inject AAV, practically every time, unless you're using certain routes of administration, in particular in the eye, you will induce immune responses against AAV. And this can lead to not only the existence of a humoral response, but toxicity. And so you can have the destruction of capsid-containing cells, loss of therapeutic efficacy because you lose these cells, but also you cannot redose and re-give again the same AAV. And of course, we're talking about also the gene transfer here and the transgene that can be expressed following AAV gene transfer. And it's clear that in preclinical studies, and in some cases in humans, you can have the induction of T-cell responses against transgene. So there's, again, also a possibility for loss of therapeutic efficacy and inflammation toxicity. So let's discuss first this first problem, which is really the pre-existing antibodies to AAV. So they are arising from the natural exposure to AAV in early life. These neutralizing antibodies can really block gene transfer efficacy, and low titers, only titers 1 to 5 or 1 to 10 are sufficient to completely block gene transfer. So these neutralizing antibodies interfere with the entry of the particles and block their binding to cellular receptors, but they also interfere in post-entry mechanisms They will also potentially amplify and make worse antibody-mediated phenomena such as phagocytosis or antibody-dependent cytotoxicity, as well as complement binding and activation. And I think you'll see that this is very important. And we don't know really how much this pre-existing immunity seems to correlate with the amplification of problems, but it looks like it's really problematic. So what solutions do we have? First of all, the first one is just to screen and to eliminate seropositive individuals. So you just have to eliminate certain population of patients that you could treat. You could also try to use modified capsids or develop self-capsids, and some people are in fact trying to modify these different residues, but the problem is that the residues that are used for entry into the target cells, which determine basically your efficacy, are also very often those that determine their interactions with immune cells. So it's really complicated. People have tried many different ways to eliminate these responses. One is to try to deplete B cells that produce the antibodies, for instance, with rituximab, but this is not sufficiently effective. And in fact, antibodies have a very long half-life and they stay in the tissues, and you can remove B cells, but the antibodies are still there. So you can try to remove antibodies by plasmapheresis, and some of my colleagues have demonstrated that multiple rounds of plasmapheresis can in fact lead to a point where you can then overcome and inject your AV, but this is clearly not something that you could apply in a large population. It's been suggested to use capsid decoys to try to deplete, but this is again something that's probably not a very good idea, because you generate a lot of T-cell epitopes for the capsid presentation, and also it's possibly a possibility for complement activation. So my colleagues at Geneto in Federico Mingozzi's group have in fact tried to develop a new strategy to eliminate those antibodies. One is to use imiflidase, which is also called IDS, IgG degrading enzyme, which is derived from Streptococcus pyogenes. So it's an enzyme that will cleave specifically IgG. And they have shown in mice, but also in non-human primates, and they've published this recently, that you can in fact inject this IDS and completely reduce the pre-existing antibodies, and then be able to administrate your AV vector. So this is a very promising strategy to be able to in fact include patients who are already seropositive into clinical trials of AAV gene therapy. Now, you also have something that happens systematically, is that when you use AAV, then you induce immune responses against the AAV capsid. So how it works is that you have, if you can avoid the, of course, the pre-existing antibodies, but you bind your AAV to receptors. This includes in particular TLR2 also at the surface of the target cells, but there is an entry of the AAV into the cells, and then there's exposure of a certain number of motifs, for instance, the DNA that's recognized by TLR9, but also you have the endosomal escape, degradation of the capsid, and presentation of the peptide epitopes to T cells, and generation of immune response. So most people here, like in this schema, are very focused and very concerned about CD8 T cell responses to the capsid, because these T cell responses are effector cytotoxic T cells, and they will kill the target cells. But, and this clearly was shown by Federico Mingozzi a long time ago already, when they did the clinical trials with CATI-HI, with the factor 9 clinical trials, where they found that they had induced the capsid T cells, CTLs. And clearly this was found again in hemophilia clinical trial by Amit Natwani, a little bit more recently, where he could correlate at the same time the liver toxicity with transaminase elevation with the appearance of capsid-specific CTLs. So this is really problematic, of course, and it can be controlled by corticoids. And now, in fact, most of the clinical trials are systematically using corticoids either prior or during treatment to avoid this induction of capsid-specific CTLs. But I'd like to get back to the antibodies, because I think this is something that is really problematic and really difficult to control. So again, AAV administration will induce anti-capsid serotype neutralizing antibodies. It will prevent re-administration. And really, this is something we need to address because we are starting to use gene therapy with AAV in children, and it's most likely will require second or third doses to prolong the effect of gene therapy throughout life. So this is really an important problem that needs to be overcome. So you can, of course, try to go very hard and try to block the source of the induction of antibody, which is T cell responses that then secondarily activate the B cells. And so it's been shown, in fact, a long time ago by some of my colleagues at Geneton, and you know very well, Luis Garcia, for instance, Stéphanie Laurent, that you can use CTLA-4 Ig or anti-CD40, and others have also used cyclosporin A and non-depleting CD4 antibodies to really block this T cell response and prevent the induction of anti-capsid T cell or B cells. Now, of course, unfortunately, these very strong immunosuppressive approach are very risky. They have secondary effects, and the risk-benefit ratio is not always acceptable. So it's very important to find non-toxic alternatives that can be implemented to prevent this induction. So again, IDES has been shown to, in fact, be a solution. You can completely cleave the antibodies as you induce them. It's been shown in this same article here that in mice or in non-human primates, you can, in fact, prevent the induction and allow the redosing with the secondary antibody, secondary AAV. There's another alternative also that was shown by Federico Mingozzi and colleagues, which is to use nanoparticles that encapsulate rapamycin. So they are used at low dose and it's not toxic, and they can be used in mice or in non-human primates, again, to block the induction of antibodies to the AAV8 capsid and to enable the redosing and facilitate transduction. So this effect is clearly blocking the activation of the T cells, but perhaps also inducing the population of Tregs in this context. We've also in my lab tried to look at some of the mechanisms involved in this induction of anti-capsid antibody immune responses. And we come across something that was completely unexpected, in fact, by using a anti-inflammatory compound, which is called MNTBAP, which is a superoxide dismutase mimetic. We found, in fact, unexpectedly that this drug can downregulate CD4 in a temporary fashion at the surface of T cells. So this is the work of Timian Nieldes in the lab. And we found that, in fact, by reducing temporarily CD4 on the T cells, that we could, in fact, overcome this period of priming and activation of the CD4 cell in the mice, and then administer the AAV vector and avoid the induction of antibody and enable the redosing. So this is unfortunately not a drug available, but this is a concept that's very important. And I think that when people are in fact trying to schematize what's going on in the immune responses against AAV, they completely forgot this help and the induction of CD4 cells. And I think that this is probably the most critical and important arm on which we should really focus our attention in terms of immunosuppression and immunomodulation, because clearly professional APCs will capture and will present the capsid peptides to T cells, but they will also activate in a separate way the B cells. And this antibody production here will be linked then and will co-amplify what's going on in terms of crosstalk between the CD4 cells and all of the other cells of the immune system. And you will also have this phenomenon of opsonization, inflammation, and of course, complement activation. And this problem of complement activation, I put here the complement because this is an old system that has been really, really overlooked in gene therapy. And clearly, antibody-mediated complement activation is something that really should be looked much more carefully. And in fact, indeed, it has been observed in clinical trials recently that there's been some problems with complement activation. So for instance, one of the trials in Duchenne muscular dystrophy by Solid Bioscience, it was an AAV systemic delivery for a microdystrophin gene transfer. They have treated their first patient with a dose escalation trial, but when they reached the high dose in some of the patients, they observed severe effects with low platelet count, red blood cell reduction, and also some of the symptoms that act and demonstration that in fact had a very strong activation of the complement system. So the trial was put on hold, and it has resumed. And to overcome this problem, they've had to improve their manufacturing to have better quality AAV particle with less empty capsid, but also they're using prophylactic anti-complement inhibitor, the eculizumab, and a C1 esterase inhibitor directly into their trial, and they're increasing the dose of corticoids in the first months post-dosing. So that shows you how serious and real these problems are. And again, this complement activation was really overlooked. Now, of course, we know that we can induce, in fact, complement activation when you use nanoparticles. So this is a recent paper that came out in Frontier Immunology, where they describe, in fact, the induction of complement in the context of cancer nanomedicine. So in fact, this could be useful in this context. But I would like to also cite Aurélie's work, who has identified, of course, that certain type of nucleic acid sequence can, in fact, induce also complement activation, even though in this case, the response was very rapid. So this is probably a different mechanism than what we've shown before, that is probably at least partly linked to antibody-mediated activation. So I will wrap up because I think, but I would like to say that there are many other immune problems, one of which is in fact the induction of transgene-specific immune responses. And this has also been observed in clinical trials, here illustrated, for instance, in MPS IIIB clinical trial, where following injections of the AAV into the brain, they have in fact generated anti-transgene immune responses. We know we can control them by regulating, again, the presentation of the transgene to professional APCs, but you can control that very well in normal model mice, for instance, but not at all in mice that have, for instance, dystrophic phenotypes, such as these mice, sarcoglycan deficient mice. So there's this problem with indirect transgene presentation that is very, very complicated to control in the context of inflammation. So just to wrap up, there's probably no ideal AAV vector at this point, even though people should really try to improve it to escape pre-existing neutralizing antibodies, to minimize the induction of anti-capsid response, and to prevent also the presentation of the transgene to T cells. But the problem is seen also with other vectors. Lentiviral vectors are very immunogenic. I can show you this data in mice. Cas9, also people are using CRISPR-Cas9. There is pre-existing immunity to Cas9 protein. So this is a broad general problem, not just with AAV. So I just want to wrap up and summarize and say that AAV gene therapy is a very well-tolerated and well-used and useful approach for gene therapy. But these complex immune responses that can alter its safety and efficacy still remain problematic and need to be better studied and understood and controlled. So now people are systematically using immunosuppressors or immunomodulators. I think there are promising advances to try to find better products that can have low toxicity. And so I think this could benefit the whole field for all of the different types of approaches. I think people really overlook the patient status, whether it's the genetic tolerance to the transgene or the inflammation. I think immunomonitoring also is very important to really understand what's going on. And the immunogenicity, intrinsic immunogenicity of the product also should be really taken into consideration. And the last point, and then I'll stop, is to say that unfortunately, there are very few good models, if any, of these immune responses. And often you have to go into man to understand what's going on. So we need new models of human immune responses. I think it's really urgently needed. So thank you to all my colleagues that I've used data from, and thank you for your attention.